dear students, steps in DNA fingerprinting. How is DNA fingerprinting done? This topic pertains to genetics and evolution in your biology subject of class 12. One of the important techniques used in this DNA fingerprinting is VNTR, variable number tandem repeats, or Southern. This is done by the Southern blotting technique, in which radio-labeled VNTR acts as a probe. This acts as a probe in the DNA fingerprinting, what we call as variable number of tandem repeats. There are a large number of steps. How do we proceed in DNA fingerprinting? As I have already told you in the previous lecture, that this DNA fingerprinting is a very important technique in the detection of crimes in our society by the police, where the court cases are referred, where the forensic laboratory plays a very important role in this application of the technique to detect the parental disputes, to solve the parental disputes in court cases, to solve the criminal cases in our society. The steps which are used in this DNA fingerprinting are: number one, for DNA fingerprinting, DNA is isolated from any body cell or even from blood stains, semen stains, or hair roots. DNA can be obtained by a special probe from the body cell or from the blood stains of that very person, which is suspected. Blood stains or semen of that very person who is a criminal or the person under investigation. DNA can be obtained. DNA sample is first digested with a restriction endonuclease enzyme, and the digested sample is then get electrophorused. A restriction endonuclease enzyme is used as a molecular scissor to cut this DNA into several fragments. And then the digested sample of DNA is subjected to electrophoresis. Electrophoresis is a technique of separating the substances based upon their polarity in an electrolytic medium. This is the second step, in which the restriction endonuclease or the molecular scissor is used. Then the third step involved in this DNA fingerprinting is DNA bands in the gel are denatured into single strands by treating them with alkali solutions. In the gel, DNA strands are made then single-stranded. They are denatured into single-stranded. The DNA is always double-stranded. That can be made single-stranded whenever the bonding between adenine, thymine, and guanine cytosine is broken by a special technique. The fourth step involved in this is gel is laid on the top of a buffer-saturated filter paper, placed on a solid support that is called as glass plate. The glass plate is used where this gel is placed, in which contains buffer-saturated filter paper, with its two edges immersed in the buffer. The two edges are immersed in the buffer solution. The fifth step involved in this is a sheet of nitrocellulose membrane is placed on top of the gel, and a stack of paper towel is placed on the top of the membrane, which acts as an adsorbent tissue. Due to the process of this adsorbent tissue, the adsorbent tissue can be utilized, which is nitrocellulose membrane. Used as an adsorbent solution. There are further steps. A weight of about 0.5 kilograms. A weight of about 0.5 kilograms is placed on top of the pack of paper towel. Any kind of weight, whether iron or anything, that is placed on the top of that very towel. Leave this arrangement for a few hours or overnight. The buffer solution is drawn up by the filter paper wick. The filter paper wick, which is dissolved in the buffer solution, that absorbs the solution. through the gel to the nitrocellulose membrane and finally to the paper towels. While passing through the gel, the buffer carries with it single-stranded DNA molecules. This buffer carries with single-stranded DNA molecules, which are not double-stranded. While passing through the gel, the buffer carries with it single-stranded DNA molecules, which bind to the nitrocellulose membrane. While the buffer passes through it to the paper towels. Next day, paper towels are removed and discarded. On the next day, the paper towel, which is removed because it has no utility, then there is one more step in this. How do we conduct the DNA fingerprinting? The eighth step involved in this technique is nitrocellulose membrane with single-stranded DNA bands is incubated at 80 degrees centigrade. These single-stranded DNA threads are incubated at 80 degrees centigrade for 2 to 3 hours to fix the DNA bands permanently on the membrane, so that the DNA band is fixed permanently on a membrane. There is one more step. This membrane now has a replica of DNA bands from agarose gel and can be used for hybridization with specific labeled DNA or RNA probes. Then there is one more step. The membrane may then be washed to remove any unwound DNA, and X-ray film is exposed to the hybridized membrane to get autoradiograph. Lastly, the autoradiograph is obtained on the X-ray membrane. Autoradiograph of this single-stranded DNA threads. This technique has a very significance. The technique of DNA fingerprinting, which we were discussing for a few minutes, has a very significance. DNA fingerprinting shows polymorphism of DNA. It shows polymorphism of DNA, which is used for identification of a person with much more certainty that has hitherto been possible through techniques of blood groups. Since the number of blood groups available becomes a limitation, the technique has shown the possibility of two persons having same pattern of DNA fingerprints is very remote, exception monozygotic or identical twins. This is the significance of DNA fingerprinting, which is used in the forensic science laboratory today.